Thank you, Alan, for the kind introduction. Thank you for the organizers. I really appreciate the opportunity to be here and to share with you our data. And it's kind of a collection of data over the years that evolved from what was originally a project with the Office of Navy Research. And that evolved to a number of projects. I'll be focusing a lot on cancer, on the metabolic management of cancer with the ketogenic diet and hyperbaric oxygen. A show of hands, who was able to attend the last seminar with Beth, Zupekania? Okay. And who is not familiar with the ketogenic diet? Okay, thank you. So most people, but so some of what I'll be going over is a review of Beth's education, and I will kind of get into how we have further developed technologies that can allow us to mimic the therapeutic effects of the ketogenic diet without necessarily the highly restrictive part of the diet. So, let's see. On this side. Okay. I always like to begin by thanking our sponsors. So the research that we do that I'm showing you today really cannot be possible without the help of our sponsors. The Office of Navy Research was the original kind of government organization that has funded me for the last 10 years to develop various technologies that can allow us to understand the therapeutic effects of hyperbaric oxygen and also the damaging effects of very high levels of oxygen that a Navy diver could experience. The Department of Defense has been able to fund the development of a lot of the technologies that I'll be able to show you. Scivation and Quest Nutrition, you've experienced some of the Quest foods out there in the hallway. They have been very supportive of, prior to me doing this research, they didn't know anything about the ketogenic diet, and they came on board. They recognized this area of hyperbaric oxygen and the ketogenic diet as having potential therapeutic effects, and now they're developing food products, even pre-packaged whole foods in addition to snack foods. And these products will make the ketogenic diet more palatable and actually offer an increased compliance for patients. And I work with a number of foundations, including the Glucose Transporter Deficiency Foundation and Winning the Fight. This is primarily an ALS and neurodegenerative disease. GLUT1 deficiency, just for your information, is a rare metabolic disorder where there's a deficiency of the glucose transporter. So the ketogenic diet is actually the standard of care for that. So you may recognize the ketogenic diet as, some people think of it as a fad diet, a high-fat kind of fad diet used for weight loss. But the history of the ketogenic diet is very fascinating. It's been used for about 90 years for the management of epilepsy. So that's how I got turned on to it, studying oxygen toxicity. So before I even started studying this as a PhD student, my PhD research was really focused on human physiology at environmental extremes. So what happens under low oxygen? We call that hypoxia. What physiological changes, what molecular biological changes happen at low levels of oxygen? So we would study, I specifically studied the brainstem mechanisms that controlled our respiration and our heart rate. So the neural control of our autonomic regulation and all the signaling processes in the brain that sort of was associated with how we adapt to those environmental extremes. When I finished my PhD and went on to my postdoctoral fellowship, that's when I connected with the Office of Navy Research, and they were very interested in the effects of high pressure oxygen, specifically mitigating the dangerous effects of high pressure oxygen. So that ultimately led me to the ketogenic diet, interestingly enough, and I'll talk how that all happened. But to study the effects of high pressure oxygen, we needed to develop very specialized technologies that allow us to actually go into cells or visualize what happens at the level of the cell membrane and at the level of even the mitochondria underneath the cell membrane. So I have some images of cell membranes and the mitochondria within cells, which are the powerhouse. They make the energy that the cell uses. So we've actually developed these technologies and installed them inside a hyperbaric chamber. We worked with Reimer Systems on that. So this is one of our larger chambers, actually, and it looks at the smallest thing. It looks at things at the level of nanometers. So its name is the atomic force microscope, and it uses a scanning probe that goes across the surface of the membrane, and these probes are very sharp, down to the level of one atom. They're monoatomic. And they can, it allows for the resolution of the cell membrane in a way that we can determine how the cell and the underlying cellular components change as a function of oxygen and a function of pressure. So another chamber that we have is to do radiotelemetry. So we have our rodent models, our rodents get inside the chamber, and we have a video camera system in here, and we can measure brain activity. We can measure EEG, we can measure diaphragmatic EMG, which is respiration, body temperature, activity. We get all these physiological parameters come up on the computer screen, and then we simulate what a Navy SEAL would do when he does an oxygen rebreather dive. And we see how the physiology is changing, and we understand how the brain's activity is changing under those environmental extremes. So these technologies allow us to kind of quantify and understand the problem, and they later play an important role in screening various technologies so we can look at brain activity with and without, the state of ketosis. I started studying all this looking at anti-seizure drugs. And I realized that the drugs offered help, but diet was actually more powerful than drugs. And the ketogenic diet is actually used clinically for drug refractory or drug resistant epilepsy. So when drugs fail, the diet is used, and Beth might have talked about this in the last talk, and in about 2/3 of the cases, it can be used to manage seizures. So I was fascinated with that. I didn't understand that when I started studying this. So our research on CNS oxygen toxicity sort of evolved and led to developing metabolic therapies. Some of them are ketogenic diet or ketone supplement therapies for a wide range of pathologies, including neurological disorders, Alzheimer's disease, Parkinson's, Angelman syndrome. I talked about glucose transporter deficiency syndrome. So these are just some of the projects that we're doing. We have a wound healing project that's really exciting, showing that if you're in a state of ketosis, it enhances blood flow to wounds that are ischemic, and it also lowers the inflammation there. A good chunk of my talk in the last section will be talking about our results using these approaches to manage cancer. So CNS oxygen toxicity, for those that don't know, results from breathing oxygen at a very high level, above the level that you would typically use, that you would almost always use for hyperbaric oxygen therapy. So in hyperbaric oxygen therapy, the very top level you'd use is 3 atmospheres of oxygen. And once you're at 3 or even above 2.5, the military is kind of conservative, you can potentially experience CNS oxygen toxicity if you're down long enough. And it's a limitation of hyperbaric oxygen therapy, and we know that's used for chronic wounds, it's used for decompression sickness, if you have too much nitrogen in your body, if you're diving, carbon monoxide poisoning, a wide range, about 14 to 15 different FDA approved applications, but a growing and emerging number of off-label applications for hyperbaric oxygen therapy are evolving all the time. So it's a very exciting area to be in, to specialize in. So CNS oxygen toxicity is a limitation for special operations diving. In particular, Navy SEALs use a closed circuit rebreather. And so the air that they're breathing out is recycled back, the carbon dioxide is taken out, and it's essentially 100% oxygen. And The advantage to that is that you avert the potential for narcosis or decompression sickness, but more importantly, there's no bubbles coming up, so there's a stealth component to it. So you can be in a lake and be completely silent underwater. The disadvantage is that if you go down just 50 feet of seawater, within 10 minutes, you have the potential for having CNS oxygen toxicity. If you were to dive down, for example, down to 130 feet of seawater, that would be 5 atmospheres of oxygen, and you would likely have a seizure within 5 to 10 minutes, like almost certainly. So it's a severe limitation. You know, you talk to these guys that do their operations, their missions are such that sometimes they have to swim across the bay for up to 5 to 6 hours and then dive down deep to check something out, to plan a mine, to do something. And they often go past these limitations. So that could be critical to the mission, that can end the mission and certainly end their life. So the question is, why do these seizures occur and how can we prevent them? Right, so these are, I talked about the methods. One is atomic force microscopy, fluorescence microscopy, laser scanning confocal, electrophysiology, and radiotelemetry. So this allows us to visualize at the level of the molecule to the level of whole animal physiology, including the measurement of free radicals. We're all familiar with reactive oxygen species, also known as free radicals. So what we've done in our lab is develop technology to watch in real time the formation of free radicals within the cells. And this gives us a lot of insight into how hyperbaric oxygen impacts the cell, because in some cells the production of free radicals is relatively small under hyperbaric oxygen, and in other cells they spew out massive amounts of free radicals. And that would be cancer cells. And that's something that I observed early on, and I'll talk a little bit more about later. So we've adapted these techniques for use in hyperbaric chambers. And I showed this to you and that would be, this is actually the transmitter that's implanted inside the rodent model that we use. So we do surgery and we implant this transmitter, and it measures EEG, so it measures brain activity, diaphragm activity, and heart rate activity, and it also measures activity, general movement activity, and temperature. And when it's implanted, it transmits to a receiver that's wired through the wall of the chamber here. So the animal is freely moving, there's no wires connected to them, and we have a very good kind of overview of their physiology and how that changes over time. So using those techniques, we've demonstrated that under high levels of oxygen, that or relative changes in oxygenation causes an increase in a free radical called superoxide anion. And in the presence of nitric oxide, can form peroxynitrate. In the presence of an antioxidant that your body makes, that can break it down to hydrogen peroxide, and that can ultimately go to water, or in the presence of iron in certain tissues, it can transform into the hydroxyl radical, which is highly reactive and can cause tissue damage. So we've developed technologies to measure these in real time. One of my postdoctoral work was actually developing methods to measure superoxide anion in the hippocampus. So the hippocampus is an area that plays an intimate role in learning and memory, and it's very oxygen sensitive. And we think that CNS oxygen toxicity seizures, the locus of the seizure, may actually be generated from this region of the hippocampus. So that's why I studied it. And if we zoom in on the cells, we can start to see under really high levels of oxygen, these punctate staining is the mitochondria. So the mitochondria are actually the source. The metabolism of the cell is the source of the free radicals. These reactive oxygen species and reactive nitrogen species contribute to accelerated lipid peroxidation, protein oxidation, protein nitration, and membrane damage. The surface of our cells have a 5 nanometer thick plasma membrane that's a composite of a wide range of fatty acids. So in the brain, you've heard of DHA, which is docosahexaenoic acid. It's very rich in fish oil. So when you're eating fish, a lot of those fatty acids can get incorporated into your brain, and your brain loves fish for that reason. It's a major part of the cell membrane. The more fluid we keep the cell membrane, essentially the more healthy our brain cells are. So we also do a variety of electrophysiological techniques involving measurement of calcium homeostasis, and the level of calcium in the cell and the release of calcium plays a really big role in the cellular excitability. So there's various pharmaceuticals that I was looking at targeting the calcium, trying to buffer that increase in intracellular calcium. But if the metabolism is the root of the cause, this is more or less like a downstream epiphenomenon. So targeting energy metabolism is basically targeting the mitochondria, where the free radicals are taking place. A lot of what we're doing now with epilepsy and other disorders we're targeting downstream effects of the initial kind of problem, right? So we also do electrophysiology, where we can stick an electrode into the hippocampus and measure all the neurons firing together. And that's called, the recording would be, the measurement we make is called an orthodromic population spike. If we measure here and stimulate here, and we look at the amplitude of that spike, that gives us information of how excited that tissue is under certain levels of oxygen. So the higher the level of oxygen, the greater the spike is over time. And then under conditions of hyperbaric oxygen therapy, or hyperbaric oxygen to the level that would cause CNS oxygen toxicity, we see these secondary spikes form, and that's analogous to a seizure. And at that level of oxygen in our in vivo model, or a whole animal model, we see seizure-like activity in the rodent models. They'll get back on their hind limbs, and they kind of freeze and shake, and that's quickly reversible if we flush in air. Once we see that, we quickly reverse the condition with air. So kind of a summary of that, and just a snapshot of what I described, was that high oxygen pressure essentially stimulates metabolic dysregulation, which is a result of, or results in oxidative stress. Oxidative stress contributes to the metabolic dysregulation and vice versa. And this contributes to the seizures. The metabolic dysregulation kind of impacts, from a global perspective, the activity of the brain and the communication from one brain region to another. And we're starting to get a greater understanding of what brain regions may be most impacted by high levels of oxygen. And that can give us insight into kind of what metabolic substrates and even kind of what drugs we can also use in tandem. So the strategies that the military has used to prevent oxygen toxicity include antioxidants, anti-epileptic drugs, oxidative preconditioning. So if you expose someone to hyperbaric oxygen therapy and you do it several times, their antioxidant capacity is upregulated. So they're actually better capable of dealing with the stress. But we're working on protocols now that would define the oxidative preconditioning. But it tends to confer a relatively small advantage compared to some of the high-dose anti-epileptic drugs, or what was really interesting to me at the time I started looking at therapies was starvation. So prolonged fasting or starvation could delay the latency to seizure by over 200%. And that was pretty much more than most of the anti-epileptic drugs that I was looking at. And this was confirmed in several labs. So I became interested in how does brain energy metabolism change under a state of prolonged fasting. And much of what we know about starvation in the brain comes from the work of George Cahill. He was at Harvard Medical School, and he did a fascinating study where he starved his subjects for 40 days. And his subjects were mostly medical students, a few residents. And obviously, this could never be reproduced today. It would never pass IRB. I don't know, the IRB must have been on vacation or something, but they fasted them, they gave them no food, just water and electrolytes for 40 days. And what they saw is that ketones, the ketone bodies I'll be talking about, are essentially zero in a fed state. So, but after about a week to 10 days of fasting, the levels of ketones in our blood actually exceed the level of glucose. So your glucose will come down, but then it kind of stays stable because our liver is very good at gluconeogenesis. So the amino acids that are in our skeletal muscle, and also the glycerol backbone of the fatty acid. So fats, triglycerides are made up of glycerol and fatty acids, and part of the fat molecule can convert into glucose. So after about 10 days, he did an elegant study where he looked at blood flow going to the brain and coming from the brain, and determined that approximately 2/3 of the energy that the brain was using was coming from ketone bodies. And that prior to this work in 1967, it was thought that glucose was the exclusive energy source of the brain and that no other fuels could be used. I became kind of interested in this study and actually fasted myself for 1 week and was able to get the glucose. This is milligrams per deciliter, but in millimolar, it would be similar to about this level right here, the metabolic zone. And that metabolic zone, being in the metabolic zone, is very therapeutic for a number of different disorders. For the metabolic management of epilepsy, in an organ system or a physiological system, it's very difficult for cancer to grow if you're in the metabolic zone, because you're suppressing glucose availability, you're suppressing the hormone insulin. So your insulin needs to be essentially zero to make this amount of ketones. And we know that cancer cells grow tremendously well in the presence of insulin. So you have the absence of insulin, you have very low glucose, and your ketone bodies are elevated, which I'll show you have anti-cancer effects. In an extension of the study that definitely would not be approved by IRB and probably not approved by an animal care and use committee for even rodents, he demonstrated that an injection of 20 IUs of insulin into his subjects that were fasted caused a profound decrease in blood glucose, down to the level of almost 1 millimolar. So he suppressed their glucose levels down to 1 to 2 millimolar. We're talking about 20 milligrams per deciliter, 18 to 20 milligrams per deciliter. So this caused severe hypoglycemia in his fasted subjects. His subjects all survived without brain damage, and they were essentially, their brains were adapted to ketones. And what was very interesting, they were asymptomatic for hypoglycemia. Even though they were in a level of hypoglycemia that's universally fatal, they weren't experiencing any signs of hypoglycemia, and they even took some cognitive function tests and performed remarkably well. So I teach the medical students, and this is kind of from some of the slides that I have, just showing the pathological effects of hypoglycemia. At this level of hypoglycemia, it almost surely causes a coma and, interestingly, a seizure, because you're de-energizing the brain that has no glucose and no ketones, so it like basically depolarizes. So it's a very graphic demonstration that ketones can replace glucose as an energy source. So I'm not going to get into all this, but the steps for glucose to make energy really depend on getting glucose into the cell, and it involves this transporter and also this enzyme called pyruvate dehydrogenase complex. And in conditions of Alzheimer's disease or traumatic brain injury, there's an internalization of the GLUT3 transporter and an inactivation of this enzyme. So what's interesting is that ketones just kind of blast right through the membrane and go right to the Krebs cycle to make energy. So the ketones produce these reduced intermediates that drive the electron transport chain. It's how our cells make energy. And ketones do it essentially in a much more efficient manner. We know that nutritional ketosis mimics the effects of fasting, so by elevating ketones. Instead of our body converting fatty acids into ketones, we can develop a diet that's primarily made of fat, and Beth talked about this at the last seminar. When we follow a ketogenic diet, we're basically feeding our body fat and forcing our body to burn fat and ketones instead of carbohydrates and sugar. And we also liberate more fat from our adipose tissue. To do this, we need to basically break down a lot of the stored sugar in our liver, called glycogen, and suppress the hormone insulin. And when we do that, our liver generates these ketone bodies, which are like water-soluble fat molecules that can easily get into the brain. And when we have these molecules elevated in our blood, they're highly efficient sources of energy for our neurons and our astrocytes in our brain, and also our heart. The heart runs about 25 to 30% more efficient off ketones than it does off glucose. And that was shown years ago. And now we understand that ketones can also be a source of energy for the skin. We do experiments in human dermal fibroblasts in our lab, and we can give glucose and ketones, and we can take away the glucose and give pure ketones, and the cells survive really well. Cancer cells will die when you do the same manipulation. And ketones can also be a source of energy for skeletal muscle. This is very difficult to sustain. So you need a trained dietitian, and you need a high level of compliance to do that. So some resources some books that I got before I started doing this research, and as I was starting to learn, I connected with Jung Roh. He was at Barrow Neurological Institute at the time, and I connected with Dr. Eric Kosoff from Johns Hopkins. So Dr. Kosoff has been a pioneer in the modified Atkins diet, so a less restrictive form of the ketogenic diet that's remarkably effective for controlling epilepsy. So the earlier version of this book, the 4th edition, was the ketogenic diets for the treatment of epilepsy. And the 5th edition is now called the ketogenic diets for epilepsy and other disorders. So there's a whole bunch of emerging applications from neurological diseases and now cancer that Johns Hopkins is kind of getting patients for. And even the Charlie Foundation that Beth is working for gets a lot of patients interested in using the diet for cancer. And so I kind of stepped into this research at kind of a fortunate time. See, I'm kind of stuck here again. Okay, so before I go any further, some of you know this, but I'm going to briefly go through some ketone terminology. So ketone bodies, I already told you, are essentially they're energy substrates from the breakdown of our fats. And the benefit are that they're smaller molecules and they go past cell membranes and they can energize our cells. The state of being in ketosis typically involves having a blood level above 0.5 millimolar. And this will make sense when I show you some graphs and show how the blood levels can be elevated with different types of ketone supplements. Nutritional ketosis is a dietary strategy to elevate your blood ketone levels. So when doctors typically hear about ketosis, they think ketones are bad because they're typically associated with pathological conditions, one of them being diabetic ketoacidosis. Another is alcoholic ketoacidosis. If you drink too much alcohol, especially in a fasted state, you'll experience that. So pathological ketoacidosis occurs when ketone levels are very high, up in the 10 millimolar and above range. And our diabetic ketoacidosis pretty much only occurs in the absence of insulin. So a type 1 diabetic who does not make insulin cannot properly regulate ketone production in the liver. So without insulin, your liver essentially has runaway ketogenesis. If a person is in the fasted state, like the Cahill study, and they actually injected insulin. If you looked at their ketones went down. The insulin that went into their blood actually shut off lipolysis in the liver. So ketone or insulin is very, very anti-ketogenic. So that's important to know because your insulin needs to be suppressed to make ketones. So diabetic ketoacidosis is not something you have to worry about within the context of what I'm talking about, nutritional ketosis. Keto adaptation is a time-dependent physiological shift in your body using fats and ketones in place of glucose. And it's not an all or nothing scenario. So you gradually shift to using more fats and ketones over time. And in advanced athletes, that if you look at athletes that are at the same level of performance, and you look at ones that strictly use a high carbohydrate diet and a high fat diet, you can look at the respiration and determine that the high fat diet athletes are burning anywhere from 2 to 4 times more fat than the high carb athletes. So they literally change sort of the engine of their body to burn a different fuel source. So the new technologies that we developed, which are kind of new to science in general, are exogenous ketones. So I talked about getting into a state of ketosis and staying there is very tricky. And to do that, to circumvent that dietary restriction, we developed synthetic and naturally derived ketones that can elevate blood ketones for a number of hours. So these are strategies for inducing nutritional ketosis. So the ketogenic diet, there's a number of variants to the diet. The classic ketogenic diet is really about 90% fat, which is very hard to follow. This was called the Johns Hopkins protocol. It worked remarkably well for controlling seizures in individuals where drugs completely failed. So they realized over time, there was research done at Johns Hopkins on the modified Atkins diet and showing that a more liberal use of protein in the diet makes it much easier to follow. And they have about a similar level of seizure control with a modified Atkins. A medium chain triglyceride diet incorporates a high level of, allows for a more liberal use of carbohydrates and incorporates medium chain fatty acid into the diet. So medium chain fatty acids, which can come in an oil or in a powder, can be formulated into the diet, and these are very ketogenic oils. So when you consume them, the fatty acids are rapidly metabolized in your liver and you make more ketones than if you were consuming long chain fats. Now, there's also the low glycemic index diet. So there's been a discussion and a debate in the epilepsy community and the ketogenic diet community. Is it the ketones or is it simply suppressing and controlling the hormone insulin and keeping your blood sugar under control? We know that spikes in sugar can send people into seizures. So a low glycemic index diet, which is basically eating carbohydrates that cause a very little rise in insulin and glucose, is remarkably effective for some forms of seizures. But there's many forms of epilepsy that are not responsive to a low glycemic diet, but controlled with the ketogenic diet. So these are the variants of the diet, and you have the development of ketogenic ketone supplementation, fats that are converted to ketones, or the ketone molecules themselves combined with various minerals like sodium, potassium, calcium, and magnesium, and they are formulated into a liquid or a powder. And when these are consumed and you look at your blood, it looks like you've been fasting for a day or two. So that's the benefits, that you can consume kind of these products and be sort of in a state of instant ketosis. Now the products that we originally sort of started formulating were synthetic ketone esters. And these are very powerful. They can put you in a state of starvation ketosis, not in 7 days or 10 days, but in about 10 minutes. So the advantage is that you can have neuroprotection almost instantly, and they can be given IV, they can be given orally. You're sending a very efficient fuel source to your brain, and it's protecting your brain. And we'll talk about how this can be used for oxygen toxicity. So here's some data from pharmacokinetic studies we did on exogenous ketones to produce rapid and sustained ketosis. When we did this study, we just took the blood up to 240 minutes, and we thought that we really thought that 4 hours the blood ketones would come down. So at the time, we were just taking the blood and sending it off to Case Western for analysis. We didn't even know that the blood levels were elevated at 4 hours. But this is the millimolar concentration, and the therapeutic level of ketosis is typically around 1 to 2 millimolar you need to maintain. So we were, with the synthetic ketone esters that we developed, a single administration resulted in therapeutic ketosis for a very long time period. So a human equivalent of this, to put a person into a state of therapeutic ketosis, would be about 1 gram per kilogram per day. So most people in here are between 50 to 100 kilograms. So for me, I would take about 100 milliliters of a ketone ester, and it would, that graph of the people who fasted 40 days, if I consumed that and you looked at my blood 30 minutes later, it would look like I fasted for weeks. So it almost instantly puts you into that fasted state. What's interesting, that it not only elevates, this is blood beta hydroxybutyrate, it tends to decrease your glucose levels. So even if you're eating carbohydrates and you consume it, this individual was like a 95 glucose, it suppressed his glucose down to about 70. So we do a lot of studies with metformin. Metformin, you may have known, it's like an anti-diabetic drug, it lowers your blood glucose. Exogenous ketones are far more powerful than metformin. We don't fully understand how it's suppressing glucose, but we think it's causing the cells to take up more glucose, the muscle cells. So it's helping the glucose transporter translocate to the skeletal muscle, or it's suppressing hepatic glucose output. So we think this is very therapeutic, and in our wound healing studies, the lowering of glucose seems to promote perfusion of the ischemic tissue. So we get an enhancement of the wound healing process by elevating ketones, but also suppressing glucose. So there's various technologies out there that you can monitor the level of ketones in your blood. So think about having an anti-seizure drug and having the technology available that you can measure the actual blood levels of that drug in there. And that's really what we have with ketones, because there's already existing technologies. We can look at urine ketones, there's a breath acetone strip, but the gold standard is really to look at the blood levels of ketones. And Abbott Labs makes a blood ketone monitor. I've been tinkering with the breath analyzer, and it shows various colors depending on your level of ketones, but I'm hoping that the manufacturer will develop it in a way to give you a number. So hopefully that'll come out soon. So in our studies, we use that technology, we use that particular ketone ester in our oxygen toxicity studies. And here you have 2 EEG recordings, and we have ventilation too. So this is the baseline measurements that we'll take at 1 atmosphere of oxygen. We essentially flush the chamber with 100% oxygen and take some baseline measurements. We also take baseline with air. And then we dive the rat to a pressure that simulates 132 feet of seawater, or 5 atmospheres of oxygen. And then we, after about 10 to 15 minutes, we start to see the EEG activity increase, and we always see a seizure. So there's nobody is resistant to CNS oxygen toxicity. So no matter if you're at 5 atmospheres of oxygen, everyone on this planet will succumb and have a seizure. There's no way to resist it. What's interesting is that we found that the respiration, we found some distinctive changes in the respiration that preceded the seizure. So we're doing measurements to indicate that there's a physiological sort of biomarker that can warn of an impending seizure. So we're developing various technologies that we can wear that may actually tell the diver, hey, you're approaching the upper limits of your physiology. You need to at least surface or titrate in a different gas. So when we ran the study, the same animal, and we dosed him with ketone ester, at 50 minutes, 5 times, 500% increase in time, the rats are just sitting there grooming and they're fine. So this is the EEG activity after 50 minutes, with ketone ester protection. So he's in a deep state of ketosis similar to what a human would experience with 2 or 3 weeks of fasting. So those ketones are protecting his brain from the oxidative stress of the high oxygen environment. So we saw an actual 575% increase in the latency to seizure. And most anti-seizure drugs kind of fall in this category, but you need to use such a high dose that it becomes sedating. So the ketone esters, what's really interesting about ketone esters is that their original development was for the enhancement of the warfighter, for his physical and cognitive performance. So the original esters that were developed under the National Institutes of Health, funded by DARPA and tested at Oxford University, we tested those substances, but they did not protect against CNS oxygen toxicity because they elevated beta-hydroxybutyrate but not acetoacetate. That's the other ketone. So you need the elevation of both ketones to get the neuroprotection. The ketone ester that we studied here does enhance performance and it also decreases anxiety. So we're doing some studies with that now. NASA is interested in the technology because they think it can protect against the radiation that one would experience if they go past low Earth orbit, or LEO. So if we go to Mars and back, we're bombarded with what's called galactic cosmic radiation. There's charged particles that kind of run through us, and it makes us susceptible to Alzheimer's disease and cancer. So they're interested in developing a nutrition program. The ketogenic diet is also more dense. You get more calories per gram, so it's less weight that you have to get off the ground and into space. So NASA's mostly engineers, so the engineers love it, because it's just less weight. It gives them more room on the shuttle. So these are other applications. Obviously, there's a lot of medical applications. So kind of in summary, fasting, the ketogenic diet, and ketone esters work through a number of different pathways. I'm not going to go through all this, but all the red circles are things that we've kind of confirmed in our lab. And in summary, it lowers glucose, insulin, reactive oxygen species, and lowers oxidative stress in healthy cells. And we see an elevation of the ketone bodies. We see an elevation in the GABA to glutamate ratio. So GABA is a neurotransmitter that makes us feel relaxed. And glutamate is a neurotransmitter that's kind of excitotoxic and can contribute to the genesis of a seizure. So it actually causes more glutamate to be converted into GABA. And we have higher levels of these substances that are neuroprotective. And in general, if we take an area of the brain out after you've been in a state of ketosis and we look at the energy intermediates, they're significantly higher, like 2 to 3-fold higher. So that's some of the metabolomics work that we're doing. It's really like a lot of data, so I didn't put it in here. But in general, so you have the ketogenic diet, you have starvation and the ketogenic diet that can cause your body to make ketones, which function as alternative energy substrates. It's difficult to follow, so exogenous ketones can kind of bypass the whole natural ketogenesis process and directly get ketones into the bloodstream. So when I started this work in 2009, we only knew of ketones as being an alternative fuel to glucose. But now we know that ketones has a neuroprotective and signaling roles. So there's good data, there's good publications to support the longevity, the suppression of oxidative stress and inflammation, what I talked about, the neurotransmitters, and an increase in your body's sort of ability to make these intermediates, and we call this process anaplerosis. So our body basically are converting energy into intermediates that all the cells can use. So these are the applications of nutritional ketosis. So these are the ones that are proven: weight loss, type 2 diabetes, epilepsy, and other seizure disorders. And these are the emerging applications of the ketogenic diet. And these are just some of the more peer-reviewed, higher impact journals that have featured this. And most of this Some of this are human studies, and some of this are animal work. But there's now, there's registered clinical trials on autism. There's one on Angelman syndrome that's coming out. Alzheimer's disease, Parkinson's disease. These are all things that could be potentially treated with nutritional ketosis. So in our lab, in the use of exogenous ketones, this is aside from the diet, just ketones as a supplement, these are all the projects that we're currently working on right now. So I won't run through them all, but we have some of the really interesting data is on anxiety and stress. And when we were doing our oxygen toxicity studies, when we would dose the animals with the ketones, we realized that they were very relaxed when we were handling them. And the ones that were getting water, it's almost like the study was supposed to be kind of blinded, but you could tell the ones that got the water, the control, were very anxious, like they didn't want to go into the chamber. And the ones that were in ketosis were very relaxed. And that was very interesting to us. So we're doing some studies to indicate that there's greater exploratory behavior. We use what's called an elevated plus maze, and there's better learning with a novel object recognition. So we do a variety of tests to look at that. So now stepping into the realm of cancer, I want to talk about how I first kind of got into this. In 2007 and 2008, we made our first observations in cancer cells. I was studying CNS oxygen toxicity, but we had these technologies and we wanted to study not only neurons, we wanted to see what muscle cells do. We wanted to see what fibroblasts do. And one of my colleagues had brain tumor cells from a 44-year-old glioblastoma patient. And I just cultured them as another cell to study, but I didn't know anything about cancer really. So in looking at these cells and hitting them with hyperbaric oxygen, over time, we started to see these bumps form on the surface of the membrane, which are fatty acids, right? I talked about fish oil, DHA, which is a high concentration of fatty acid in the membrane. And if I zoomed into this region, which is this region, I saw these what's called membrane lipid peroxidation. So damaged fats start accumulating on the surface of the membrane. And if I zoom in more, I start to see like the size of these lipid rafts. They're about 150 to 200, maybe 300 nanometers. And they're consistent with what electron microscopy studies showed as membrane damage. And with the microscope that we developed, we could actually look at living tissue and see this process happen. So in addition to these studies showing that high pressure oxygen was damaging the cells, in parallel, I had just become interested in the ketogenic diet and was treating primary neurons and these cancer cells with ketones. And I saw it was a small effect, but I always saw that the cancer cells were not dividing as fast in the presence of ketones. Same level of glucose, but the presence of ketones, they weren't growing as fast. And I consistently saw that. And we published these observations, but at the time I wasn't really interested in steering my research towards cancer. But it was very obvious that these cancer cells were selectively vulnerable to the high levels of oxygen that we'd give with these graded levels of oxygenation. And this is a chamber that we use. The microscope's inside here. We close this up and we have a whole control panel off to the side. So these are the neurons that are kind of look like the neurons in your brain, the cells in your brain. They send out projections and make connections to one another, and they actually form like a neural network into the petri dish. And cancer cells kind of do the same thing, but in a little bit different way. So these cells were exposed to the same level of oxygen as the cancer cells. And it was obvious that the high levels of oxygen were damaging the membrane. And we did a variety of assays looking at what's called malandialdehyde. It's a membrane lipid peroxidation product. So we did assays to correlate with that. And we did further studies to actually measure the superoxide anion that's being produced in the cells. That's the free radicals that you're familiar with. So I showed the response to hyperoxia. So these are the same cells here. Before we started the experiment, the punctate staining here, these are the mitochondria. These are individual cells here. Here's the nucleus of one cell and another cell and another cell here. And here's what happens when you hit the cells with very high levels of oxygen. So the dye that we're using is called dihydroethidium, and when the superoxide is made, it actually makes a product called the ethidium cation, and it intercalates into the DNA. And the mitochondria actually have their own DNA, and there's also DNA in the nucleus. So you can see the mitochondria are just lighting up. They're really lighting up. And if we zoom in, we can see the nucleus and the mitochondria. And this is a pretty dramatic response that I saw pretty much only in the cancer cells. It took like hours for the healthy cells to get to that level. So I'm trying to understand what I'm seeing because I have a cell type that's giving me this dramatic response. So I'm calling various cancer biologists at the National Institutes of Health and Harvard and Yale and all the big things. And they said what you're observing is sort of the mitochondria are deregulated in a way. And they named Otto Warburg. And they said, yeah, you might be looking at the Warburg effect. And the Warburg effect is actually what we use to visualize cancer when we're using the technology of PET scan. This is fluorodeoxyglucose PET scan. It's a radio-labeled glucose molecule, and it's almost the gold standard for looking at the aggressiveness and the location of cancer cells. And here's an example of someone with metastatic cancer. So it's an indication that the cancer cells are outcompeting the healthy tissue for glucose. They have rates of glycolysis that are 200 times higher in aggressive cancer cells than in normal healthy cells. So I found it interesting because when I pulled my biology of cancer book off my shelf and started looking through it, there was no mention of the Warburg effect. There was no mention of cancer cells being metabolically different than normal healthy cells. But even though all oncologists would use a PET scan as sort of like the gold standard to identify, to show the metabolic differences between normal cells and cancer cells. But there's no mention of this in the biology of cancer, which is the book that anyone taking a course in cancer would have to take, and it was written by Hanahan and Weinberg, who believe that cancer is a genetic disease, right? So this was this is the figure from their paper, The Hallmarks of Cancer, which all cancer students, even medical students now have to read, and it describes the hallmarks of cancer. You have sustained proliferative signaling, evading growth suppressors, activating invasion and metastasis, so cells moving from one tissue to another, enabling replicative immortality, inducing angiogenesis, and resisting cell death or apoptosis. So these cancer cells had all these hallmarks, but it did not mention that they were metabolically different. There was some in 2011, they sort of changed their mind after some metabolic researchers were saying that this is a huge hallmark of cancer. It needs to be recognized. And now they're saying that it's an emerging hallmark. So a revision of the original paper is, and they don't even mention Otto Warburg. They just call it deregulated cellular energetics. And we know from work that we're doing now that this is a result of mitochondrial dysfunction. When the mitochondria are dysfunctional, the cells default to sugar metabolism. And that becomes a hallmark of their cellular biology. And that deregulated metabolism causes genetic instability, which causes mutations in the genome. And that causes cancer cells to be more aggressive. So the next speaker is Dr. Thomas Seyfried, and he will be addressing this question: is cancer a metabolic disease? Right? So I'm not going to steal Tom's thunder because he has a very elegant kind of presentation on cancer as a metabolic disease. Tom is a collaborator and a colleague of mine. We co-authored this paper, Cancer as a Metabolic Disease, the Implications for Novel Therapeutics. It's open access. It's in a fairly high impact, peer-reviewed journal, and it kind of covers this topic pretty well. Tom's book is available on Amazon and is a very kind of a lengthy display or review of all the literature supporting cancer as a metabolic disease. And in this article, there's actually discusses the use of hyperbaric oxygen in combination with a dietary approach. So Tom has worked out really nice diagrams in his book. This particular diagram is in this review too. So hyperglycemia and tumor hypoxia, low levels of oxygen, drive the Warburg effect. So it makes the Warburg effect even more pronounced. And it kind of contributes to it, and it makes it even more aggressive. And we know that diets that are high glycemic, high in carbohydrates and sugars, increase our risk of cancer. If we follow this over time, if we take an animal model or if we take humans and give them a high glycemic, high carbohydrate diet, and then look at them 3/4 of the way through their lifespan, you're going to find more tumors. This is pretty much accepted now by research scientists and clinicians. Hyperglycemia, if you have existing cancer, hyperglycemia will lead to a poor prognosis. If we're chronically in a hyperglycemic state, we have more inflammation and we're promoting an environment for cancer to grow and metastasize to other organs. Blood glucose, we know from a ton of animal work that blood glucose is highly correlated to tumor growth. So this brings in the discussion of the ketogenic diet to exploit the effects of the ketogenic diet on cancer growth as it lowers the glucose ketone index. So it lowers glucose and elevates ketones. We call this the glucose ketone index. And Thomas Seyfried has actually patented a formula to calculate the glucose ketone index. And we think that we can develop various technologies to give us one number. And the lower the glucose ketone index, the more likely you are to suppress tumor growth and possibly reverse it. We know the ketogenic diet has an anti-inflammatory effect, and it also suppresses insulin signaling. Insulin contributes to the activation of various growth factors which cause tumors to grow even faster, including IGF-1. So if we look at a graph of tumor growth and glucose, we see that at very low levels of glucose, this is relatively low for a rat or a rodent model, tumor growth is very minimal, if at all. And if there's an increase in the blood concentrations of glucose where they're hyperglycemic, this would be a type 2 diabetic, tumors grow very fast in that situation. We also know that the Warburg effect is really driven by low levels of oxygen tumor. So as a tumor grows, its growth really outstrips its ability to supply oxygen to the cells. So you have a core that's very low in oxygen. As the biomass of the tumor expands, the blood vessels just can't grow fast enough to get oxygen to the tumor. So you get hypoxia in the tumor, and when the cells of the tumor are low in oxygen, that actually causes those tumor cells to kick on oncogenes. Oncogenes are cancer-causing genes, so the low levels of oxygen actually make the tumor more aggressive. You get a necrotic core, but as it expands, the hypoxia causes the tumor to be more aggressive. So with hyperbaric oxygen therapy, what happens is the oxygen gets dissolved into the solution. Instead of being carried by the red blood cells, if you dissolve the oxygen into the solution, the oxygen is more likely to get past, it's more likely to reverse the tumor hypoxia and hyperoxygenate the tumor tissue. And because the tumors are adapted to low levels of oxygen, when you hyperoxygenate them, they overproduce oxygen free radicals even much more, and that triggers apoptosis. Remember I talked about evading the death signal was one of the hallmarks? So a burst in reactive oxygen species will actually trigger the death signal. It'll trigger something called apoptosis in cancer cells, and we see that nicely in our cancer models. So we actually can turn on the gene and the proteins, and we see apoptotic cell death. So a project that was the dissertation project for my research associate, she went on, completed her PhD, did her postdoctoral fellowship with me, and decided to stay on. She had cancer in her family, so she was very motivated to do this project. Her project was to look at the effects of nutritional ketosis and hyperbaric oxygen to target cancer metabolism. So you have kind of a multimodal approach. You have ketones, low glucose, and hyperoxia produces metabolic stress. So the model that we used, I wanted to use a brain tumor model, but Dr. Seyfried was very adamant that I use the VM-M3 model of metastatic cancer. The cells were actually derived from a very aggressive brain tumor. And they're so aggressive that when they're implanted underneath the skin, they move to all the organs and they go to everywhere in the body, including the brain. So he said this model, there's a lot of models that are being used right now that have been cured by many different therapies. This particular model had no cure. There was no, even the best chemotherapy only extended life, maybe about 30 to 40%. So he was like, if it works on this model, then it's likely that you have something going on here with the therapy. So this model, the tumor cells were derived from a spontaneous brain tumor. They're syngenic with the VMDK mouse, which means the mouse is immunocompetent. Most of the studies done now by pharmaceutical companies and even in academia, they use a mouse that has no immune system. It's called a nude mouse, and they implant sometimes human tissue on the mouse, and it's called a patient-derived xenograft, right? And the only reason that the cancer will grow on the mouse is because the mouse has no immune system. Otherwise, it would recognize it. What's really unique about this mouse is that it actually has a real immune system, and that the cells metastasize from one organ to another. So that makes this a very unique model. It actually made the cover of the International Journal of Cancer. A simple implanting of about 1 million cells, which sounds like a lot, but it's not a lot, 500,000 to 1 million cells under the skin, causes the tumors to form. So at about the 3-week mark, that's when they succumb to the tumor burden, and we euthanize them. So what's interesting, these cancer cells, the individual cells themselves are transfected with a firefly luciferase. So the cancer cells have a gene in them which makes an enzyme called luciferase. So when they're implanted in the skin and they metastasize to different organs, and we give the mouse luciferin, it causes the cancer cells to bioluminesce very specifically. So we have so this is not a PET imaging, it's the actual cells, the tumor cells, you can see here, this is a healthy liver and a liver with systemic metastasis. These tumor cells glow in the presence of a protein called luciferin. So it makes it very easy to track the growth and the spread of the cancer over time. So the therapy that we did, we combined the ketogenic diet with hyperbaric oxygen. I talked about the model. The diet was a 3-to-1 ketogenic diet, so roughly about 75 to 80% fat. And we used a controlled diet, which is high carbohydrate, the ketogenic diet, and we had a hyperbaric oxygen group, which was 2.5 atmospheres of oxygen for 90 minutes, 3 times a week. And all the diet groups could eat ad libitum. So we didn't restrict their calories. We put the diet in there and they ate as much as they wanted to. And we had a ketogenic diet plus hyperbaric oxygen group. So we had 4 groups in this study. And the results were really dramatic. So here's the control diet, which was a high carbohydrate diet after 3 weeks. So this is 21 days after we implanted the tumor cells, and these are just representative images of each group. Here's the standard diet plus the hyperbaric oxygen therapy, the ketogenic diet, and once again, these are just representative images, and the ketogenic diet and the hyperbaric oxygen therapy at 21 days. All of the animals in all the groups, they all succumbed to the tumor burden over time, but it was dramatically delayed in the treatment groups. So the next thing we wanted to do was to combine, we were working with ketone esters in our oxygen toxicity work. Well, what if we further enhance the ketogenic diet with ketone supplementation? So that was the next study that we did. It was a combination therapy. It doubled the survival time in the VM-M3 mice. So going back to the data I just showed you, here's the standard diet, and this is a survival curve. So each drop here is an animal that was overcome by the tumors and was euthanized. And here's the ketogenic diet in blue, the hyperbaric oxygen therapy. It was interesting is that we did not get a statistically significant increase in the control or the hyperbaric oxygen group that was eating the high carbohydrate diet. So what was interesting is that they started dropping out much later, but they all started dying off at the same time. So they lived like a healthy life, and then just kind of, that's how you want to age, right? You just kind of live your life and then drop off. They lived, we didn't think anything, and then they just succumbed to the tumors over time, just eating the high carb diet. So this is the ketogenic diet survival curve. The ketone ester, we added this to a standard high carbohydrate diet, and we saw a significant increase in survival. That was another study that we published kind of in between these two studies. And this was a combination of the ketogenic diet, ketone ester, and hyperbaric oxygen therapy. That's the green here. So we had extension of, so this is the standard diet and this is the combination therapy. And even the best chemotherapies are somewhere in here. Like the lines are somewhere in here. We're talking about the platinum drugs, like oxyplatinum and or cisplatin and methotrexate. So the standard diet and the combination therapy. So what we don't know, so we had an increase in survival time that was over 100% increase in survival, a doubling. What we don't know is how these therapies would work with radiation, with different chemotherapies, with the immune therapies that are being developed now. So it's a tedious process to study each of these in isolation and then start putting them together. And we really need to titrate in what the optimal dose of the ketone ester is. We really need to understand what the optimal hyperbaric oxygen protocol is. We think we could greatly improve on these numbers if we optimize the ketogenic diet, the ketone ester level of supplementation, and if we optimize the hyperbaric oxygen protocol. So the next thing, what are we doing on time? Almost 9. We'll go pretty quick. I want to talk about practical guidelines for implementing nutritional ketosis. And these are kind of my go-to books. Anyone that enters my lab needs to read both of these books, The Art and Science of Low Carbohydrate Living, and this is The Art and Science of Low Carbohydrate Performance by Jeff Volek and Stephen Finney. Go back. This is a book written by the Ketogenic Diets for Epilepsy and Other Disorders by Eric Kosoff. So John Freeman was really the pioneer at Johns Hopkins University, and he recently passed away like 2 years ago. He was up in age, but he's the one who advanced all this. Like, I probably wouldn't be speaking here right now without John Freeman. He was an author of this book. So this is a great book. This is a really short kind of how-to guide. I think it's just like $2 or something that you can probably find online. As it relates specifically to cancer, these are two really good how-to books. The Ketogenic Diet for Cancer by Miriam Kalamian and also Fight Cancer with a Ketogenic Diet by Ellen Davis. So for patient support, I frequently refer people who don't know anything about the ketogenic diet to the Charlie Foundation. I've spoke on behalf of the Charlie Foundation or different events. I mentioned them in my TED Talk. Jim Abrams he's a Hollywood producer. He produced a number of movies, including Airplane, Naked Gun movies. He's actually friends with Meryl Streep, and Meryl did the movie First Do No Harm. You might be familiar with that. It's a movie about the ketogenic diet. So yeah, a lot of people are surprised that Meryl Streep did a movie about the ketogenic diet, and it was about Jim Abrams and his son Charlie. And it was a drama to basically get the word out that this is a grossly underutilized metabolic therapy for managing epilepsy, which is typically controlled with these anti-epileptic drugs that have horrible side effects, especially for a developing child. So when these drugs are used in a developing brain, they can impact development that have lasting consequences where the child can never be normal. So the ketogenic diet there's no problems with the ketogenic diet. So it's a grossly underutilized approach. So this is Beth Zubak-Kania, and this is Nancy Abrams. This is Jim's wife. So the Charlie Foundation has done a lot to advance the awareness and the education. I think there's about 150 clinics in the United States and about 200 clinics worldwide. So, and as you know this, the ketogenic diet is not a high protein diet. So I want to talk about the ketogenic diet versus standard diet. The carbohydrate intake is typically decreased to less than 10% of the total kilocalories. So the modified Atkins diet is kind of what I follow. I've been following a ketogenic diet, a modified ketogenic diet, for about 7 years now. So I've been in a state of ketosis for 7 years now. And I think there's a lot of benefits to the diet. I haven't eaten, I mean, the people who follow it know that, I haven't eaten in about 12 hours and I'm not hungry and my energy level's pretty good. So that's one of the benefits of the diet. Like if you're on a high carbohydrate diet, I would have having to eat, every couple hours. So there are challenges to initiating the ketogenic diet that need to be known up front for people who want to implement this. It has been difficult. My observation that people with liver cancer or elevated liver enzymes may have a difficulty with the diet because your liver makes ketones, and you need a relatively good functioning liver to be in a state of ketosis. So although there's quite a few patients that have contacted me with liver metastasis that have done pretty well on the diet. Kidney stones or renal disease, could reconsider doing the ketogenic diet. There's been a higher correlation of getting kidney stones if you're on a ketogenic diet, but studies have now shown that if you take potassium citrate or a potassium supplement, that can pretty much abolish or pretty much reduce your chances of kidney stones to nothing compared to a standard diet. Your liver makes the lipase enzyme. If you have pancreatitis, some people have a hard time following the ketogenic diet if they have an inflamed pancreas. Fat malabsorption issues, some people just do not make the necessary enzymes to break down, metabolize, and use fat for energy. There's a variety of lipid metabolism disorders, and I think there's about 10 or 12 of them that I know of. And if someone is a registered dietitian is working with 30 or 40 people, they're probably going to come across someone who has this disorder, and they're probably just people who just didn't tolerate fat over time. Gallbladder obstruction or removal. So your gallbladder makes bile that breaks down fat. A registered dietitian will say you can't do the ketogenic diet if your gallbladder is removed, but I know quite a few patients who do. And your gallbladder makes bile, but your pancreas can also make lipase and other fat digesting enzymes that can compensate for it. Various medications can interfere with ketosis. Using steroids like dexamethasone or prednisone in high doses can increase your blood glucose and make it hard for you to get into a state of ketosis. The biggest thing that I've seen, the biggest challenge would be lack of support. So if you have a couple, you know, and the guy has cancer or is battling cancer, or the wife, and they want to implement the ketogenic diet, but one person eats one way, a high carbohydrate diet, and the other person is trying to follow a ketogenic diet, that makes it very hard. You need to kind of purge your house of pretty much carbohydrates to follow the diet, unless you have a really good willpower. But most people simply cannot follow the diet unless they have support with their significant other or their family members. You really need social support for this. And there's various companies that are putting technologies out there, even an app, that will allow you to put in your food and calculate your ketogenic diet. And if you have a question, you press a button, and then you have 24/7 access to a registered dietitian who could tell you to eat that food or not. And you simply take a picture of it, and it automatically shows them their ketone numbers. You can measure it and put it in the phone. So these companies are being developed right now. Restricted food selection, people just don't want to follow a restricted diet, but I don't see it. I think it's actually, the ketogenic diet is kind of like an indulgent diet, because you can eat fat, right? You can eat butter, you can eat things that you're told not to eat. I eat ketogenic ice cream every night, like when I'm home, and it's made, you know, I use stevia as a sweetener, and it's made with coconut cream and other things. So here's an example of some of the prepackaged ketogenic foods that will be on the market, if they're not already, by Quest Nutrition. So they're actually whole foods, and there's a variety of ketogenic diet cookbooks that are out there. The pictures on the wall here are examples of ketogenic diet cuisine. So for me, I don't find it very restricting. But we do want to move to strategies for inducing nutritional ketosis that patients can comply with. So you want them to pick, you know, to work with a dietitian to pick the optimal diet that works best for them for managing their particular disorder. There's also the idea of intermittent fasting. And being in a state of ketosis makes intermittent fasting easier, because you can go fasting and really not get hungry. Intermittent fasting is sort of fasting 18 hours, for example, and only eating within like a 6-hour window, right? So basically you go without breakfast, and you get home at 5 o'clock, and you allow yourself to eat whatever you want from 5 to 11 before you go to bed. And that's intermittent fasting. And it's becoming very popular in like the CrossFit community and the paleo community. There's a guy at National Institutes of Health, his name is Mark Mattson, and he's done a tremendous amount of research on this. He's a big like long distance runner, and he's a very high level academic at the NIH. And I just interviewed him for a podcast talk, and it was really fascinating that he follows it, and it's becoming very popular. So there's various versions of the diet and also ketone supplementation, and that can be the form of medium chain triglyceride oil, powder, or exogenous ketone salts.
They're usually very poor tasting, aren't they?
The salts?
Yeah.
Ketone esters taste really bad, but they're the most potent. But there's a variety of supplements on the market. The companies would be Ketogenics, Prove It, Forever Green, and Ketocana, and they're all supplements that are on the market. I think what's really a benefit to advancing this technology for various applications is that we have commercially available tools for accessing the state of nutritional ketosis. It's like we have a substance that we can take that'll put us in ketosis, and we can titrate the substance and measure our blood. And once we do that, you know, a couple days, we realize like how much we have to take to get to a certain level. So we don't have to measure all the time, but it's handy to measure. So we know, you might be familiar with the keto sticks. You pee on them, and then it shows your level of ketosis. The breath ketone meter. This gold standard would be the blood ketone meter, which I like to use personally. And I will soon be using a Dexcom that measures continuous measurement of blood glucose. And I'm working with some companies who think they can develop this technology. It's a patch you put on your skin. You can make it waterproof, and it transmits to a device, and that device can report to your smartphone. So for the warfighter, yeah, you can basically make this waterproof too. For the warfighter, he can basically titrate in his ketones to optimize his performance or optimize his safety for a diving scenario. So these are the things. The military has a big program. It's called Instrumenting the Warfighter, and this is one of the technologies that they're interested in developing. So for human dosing, for the medium chain triglyceride and ketone powder, here's an example of kind of my ketones. In the morning, I'm kind of in a state of just mild ketosis, and if I take one dose of one of the commercially available ketone medium chain products that are on the market, I think this might have been the Ketogenic product, I get an elevation of ketones, maxes out at about 60 minutes and then starts coming down. After about 4 hours, I get back to baseline. So this is after 1 hour. So that's a pretty high level of ketosis, and that's about the amount of powder in a packet. It comes in a little packet, and you mix it up. It tastes really good. There's different flavors. There's berry, there's orange. And after about 3 hours, it comes back down to 1.1. That's still a good level of ketosis. At 1 millimolar, you're giving your brain about a 10% boost of energy. So all the brain metabolism has been worked out that about each millimolar gives you 1 millimolar will give you about 10% boost, 2 millimolar about 20% boost in brain energy. Some of that's been worked out by Stephen Cunnane. So what is the optimal level of ketosis? I think it's somewhere between 1 to 3 millimolar. And having technologies that easily allow us to measure this and stay in this zone is going to be really important. Now here's an example of management of ketosis for neurological diseases and cancer. And it's also important to suppress the glucose levels. So this was taken from our carcinogenesis paper and also nutrition and metabolism. This has evolved from information that we'll get from glioblastoma patients trying to manage the growth of their brain tumors. It's been shown that you initiate the diet, there's a rapid decrease in blood glucose. It's more so if you restrict the total calories and an elevation of the ketone bodies. But it typically takes, upon initiation, about 7 days to achieve this. With ketone supplementation, you could change that. If you initiate the diet and then add exogenous ketones, instead of 1 week, within 30 minutes, you could achieve the same sort of suppression of blood glucose that I talked about and elevation of ketones over time. So this is an example of if time is of the essence, obviously it is to a Navy SEAL warfighter, he doesn't have a week to wait to get into ketosis. He doesn't know when the mission's going to be. And this metabolic zone that we talk about is a blood level of glucose about 70 millimolar or 70 milligrams per deciliter. That's typically how we measure glucose in the United States, which would be about 3 millimolar, 3.8 millimolar, and then high ketones. So the idea is to get the glucose as low as possible and your ketones up.
It's 3.5?
That's a 3.6. Yeah, so this glucose level is in milligrams per deciliter, but would be 3.8. So just an example of kind of what patients will send me that are successfully managing their brain tumors. So what's interesting is getting back to the hyperbaric oxygen. So Thomas Seyfried will probably talk about a glioblastoma patient that was highlighted in one of these articles. So what I find interesting is that, and it hasn't been done yet, I think that the clinical trials are ongoing right now, we need to look at the effects of that therapy in combination with radiation therapy. So here's an effect of radiation therapy, no dietary intervention, but combined with hyperbaric oxygen, looking at malignant gliomas. So this is a really interesting study. The median survivals for patients with gliomas with and without hyperbaric oxygen were 24 and 12 months respectively. So when they followed, when they did hyperbaric oxygen, their survival time was essentially doubled in this particular study. So I remember reading, this came out quite a while ago, and I know there's some clinical trials that are being developed, that are being conducted right now. I don't know the status of them or the recruiting, and but it's definitely, we need to follow up on this, and we need to implement in tandem a nutritional protocol. I think we'll get remarkable results. So here's just some data that patients send us, right? So here's a type 1 diabetic patient. She's 27 years old, and here's her glucose. So this is 400 up here. and here's hypoglycemia. She's really dipping down here. So the average insulin she was using a day is 67 IUs. Her blood glucose was averaging 151 milligrams per deciliter. 66% of the time, her glucose was like in the danger zone. It was out of range. So this is before she started a ketogenic diet, and each thing here represents a day, like over time. So here's what happens when the same patient transitions to a ketogenic diet. And remember those meals I showed you previously, they're just examples of the ketogenic diet. They were some of the meals that she was actually taking. So here are her glucose numbers after following the ketogenic diet. And this is like standard. Imagine if a drug did that. Imagine, it would be like a blockbuster drug. Like if you invested in that drug, you're set for life. So the ketogenic diet is an extremely powerful metabolic therapy for managing type 1 diabetes. So if you told, if this patient went to her doctor and said, I'm using a low-carb ketogenic diet to manage my type 1 diabetes, like her doctor would just freak out and say, I'm not even going to follow you. This is dangerous. You're going to kill yourself. But she just had like pages and pages of emails about how wonderful this is. And her insulin went from 67 IUs a day to an average of 21 IUs a day. And it's not just one patient. There's like dozens of patients. And people are really into quantifying. There's this whole like quantified self, group of people, technology that are sending me data. And it's really interesting. It's so informative to me. It's great to do the research in the lab, but I learn a lot from patients like sending me data.
Is there a hemoglobin A1C change, or you don't know that?
Yeah, you know what? I need to follow up and see. Yeah, this was like her first couple weeks, but the Dexcom is really a unique technology that I think we can. It's really fun to look at the data. So I'm going to get one for myself and kind of see how I respond, to different ketone supplements too. So clinical trials. So there's been pilot studies with the ketogenic diet. This was done by Eugene Fine. He was funded by the Atkins Foundation, actually. It looked at targeting insulin inhibition as a metabolic therapy. This is insulin inhibition. His therapy was the ketogenic diet. But there's such a stigma to the ketogenic diet, he didn't even want to put it into the title of his article. So for advanced cancer patients, these cancer patients had failed the standard of care. So they did everything possible and failed, and this was their last ditch option. And I'd recommend this. It's open access, downloading this and reading it. It's really good. They only did it for 28 days. They monitored patients that were really hot on a PET scan, and they were the ones recruited into the trial. There was another trial with 16 patients, and it looked at quality of life. I don't think they adequately measured ketone levels in this study, but it basically showed that the ketogenic diet was safe, there was no side effects, and that it was an effective tool for enhancing the quality of life for patients. But these are very small trials, 16 patients, 10 here. We need funding to do much bigger trials so we get a handle on what cancers are most responsive to this therapy. And in addition to the ketogenic diet, we need to combine it with hyperbaric oxygen and ideally some cancer-specific metabolic drugs. So things that target glycolytic metabolism or even metformin we could use. I don't have time to show you, but we've done a lot of studies with metformin, and it kind of mimics the effects of the ketogenic diet. So right now we're doing studies combining them with the ketogenic. So if you go on clinicaltrials.gov, you'll actually see at least a dozen studies that on the ketogenic diet. And when I got into this research in 2008 or 9, there was none. So there's actually been a pretty big surge in ketogenic diet studies for cancer, registered ketogenic diet studies. So most of them are recruiting. A few of them have been discontinued because they weren't able to recruit. So there's quite a few, and it seems to be growing over time. So in summary, we know that ketones, I've showed you, they have a neuroprotective properties. They're energy substrates, but also signaling molecules. Therapeutic ketosis is a promising mitigation strategy for CNS oxygen toxicity, and I believe that it'll enhance the safety of hyperbaric oxygen therapy and perhaps enhance the efficacy of hyperbaric oxygen therapy for various disorders. I think for wound healing especially. Therapeutic ketosis and HBO target the Warburg effect in multiple ways. There's multiple overlapping signaling processes that are associated with that. Therapeutic ketosis has a potential synergy with hyperbaric oxygen therapy, and I think it'll enhance the efficacy of chemotherapy and radiation therapy. So the next study we just got funding for is to look at a brain tumor model and combine it with chemo and radiation therapy, the ketogenic diet, in combination with chemo and radiation therapy and hyperbaric oxygen. So a question that I get a lot every day probably is, can we lower the pressures of hyperbaric oxygen to elicit the same therapeutic effect? And I think the highest level of oxygen that's tolerable will be probably the most effective. But we need to do the studies. So I don't have enough information, and I think that level may be unique for each patient. The level that we chose was 2.5 atmospheres of hyperbaric oxygen. I do think people will get a benefit from lower levels of oxygen, but I think from what I see in a petri dish, from what I see in the lab in our animal model, it seems that higher level of oxygen really provides that hit to the tumor. So another question is, what combinations of adjuvant therapies and standard care will be most effective? So we envision a multifaceted approach where the cornerstone of metabolic management of cancer would be a metabolic-based therapy, and these should be tried first, because first do no harm, right? And it's a movie I encourage you guys to watch, and I think we need to stand by that, as physicians need to stand by that, and incorporate nutrition is sort of the cornerstone of our general health, and I think it should be the cornerstone of managing a disease process. Hyperbaric oxygen has a growing number of applications. Oxygen is a powerful drug when delivered under hyperbaric pressure, and we need to get more studies and more funding to determine what other applications, especially things like concussion, that we can apply hyperbaric oxygen therapy to. There's a wide range of metabolic drugs that are already developed and being developed that I think would synergize with this approach. We're doing that research now, and we'll be publishing that pretty soon. So nutraceuticals, immunotherapies, gene-targeted therapies, cytotoxic chemotherapies, they're used at the maximum tolerable dose. I think cytotoxic therapies should be used at the minimum effective dose instead of the maximum tolerable dose. And I think they'd be much more effective at lower doses when used with metabolic-based therapies. I think the same goes for radiation. And supportive health is really mental health. Exercise is a huge component. The gut microbiome is something that we're getting more interested in, and I think actually plays a huge role. Think about it though, when you take chemotherapy, it stops dividing cells. And what's the most, you have a lot of your dividing cells in your gut, right? Like 70% of your immune system is in your gut. It's like the interface between the environment and you. And when you go on chemotherapy, you basically destroy your gut. your mucose lining in your gut is sloughed off, and you destroy your gut microbiome. So, and your ability to absorb food in particular. So it puts you into a fasted state, but it's more or less like a toxic state that you're in. You're kind of like in a toxic state. So this is the multifaceted approach that we kind of want to work towards, but with our emphasis on really developing in metabolic-based therapies that we hope the general medical community will embrace and incorporate into their practice. So this research is not possible. I'm just kind of overseeing all this, but this is really the engine behind the lab, and we have medical students and undergrads too that contribute to this work that are not here. But we have a growing list of people that are helping us in the lab. You can see things are kind of tight in the lab right now. Here's one chamber that we do our dives in. This chamber right here goes to really high pressures. There's one, you can't see it over here, that goes up to like 58 atmospheres. We do experiments in. And we have about 5 rooms. So this is just one of 5 rooms with 2 chambers in it. But I want to thank you for your attention, and I'm here for the remainder of the time to handle any questions that you have. Thank you.